Assalamu alaikum dear students. Welcome to Dr. Roomi lectures. Today we are going to study our very first topic from cell physiology, and this is the structure and function of plasma membranes. Most organelles of the cell are bound by membrane, and these membranes include cell membrane, nuclear membrane, membranes of endoplasmic reticulum, membranes of the Golgi apparatus, and those around peroxisomes and lysosomes. Some non-membrane bound structures are ribosomes, cytoskeleton, and centrioles. Cell membrane or plasma membrane is the outermost boundary of all the animal cells. It's a thin, pliable, and flexible layer which provides protection to the cells. Its thickness is 7.5 to 10 nanometers, and it is too thin to be seen under an ordinary microscope. However, we can see it under electron microscope where it appears to be trilaminar in shape. This trilaminar appearance of cell membrane under electron microscope is because of the arrangement of molecules that make this membrane. In this trilaminar appearance, there are two dark layers which are separated by a middle light layer. The darker layers, one on the outside and one on the inside, they are made by the hydrophilic parts of this membrane, and the hydrophobic parts of this membrane, they make the middle light layer under electron microscope. Cell membrane is composed of three types of organic chemicals, and these are proteins, lipids, and carbohydrates. In terms of mass, proteins are most abundant, which make about 55% of the total mass of cell membrane. Lipids are above 40%, and carbohydrates are just 3%. Among lipids, phospholipids are most abundant, which make about 25% of the total mass of cell membrane. A phospholipid molecule is having both polar and non-polar parts. The polar part of this phospholipid molecule is made by phosphate head, and this faces the water either towards the extracellular fluid or towards the intracellular fluid. You can see here this is the polar head made by phosphate. Being hydrophilic or water loving, it is towards extracellular fluid or towards the intracellular fluid. And the tails, which are hydrophobic or water fearing, they are repelled by water, so they will be attracted towards each other and they will come towards the center of this lipid bilayer. Because of the presence of these phospholipid molecules, which make the backbone of cell membrane, the consistency of cell membrane is fluid, it's not rigid. It's just like cooking oil, but not like butter. These phospholipid molecules, they can twirl, they can vibrate and move around in the same half of lipid bilayer. Cholesterol is the second most abundant lipid in cell membrane. It makes about 13% mass of cell membrane. In a sense, cholesterol is dissolved in the phospholipid bilayer, and it contributes both to the fluidity as well as to the stability of cell membrane. Cholesterol also contributes to the degree of permeability in between the phospholipid molecules. Cholesterol prevents packing and crystallizing of the fatty acid chains, which under some conditions, for example during cold weather, can decrease fluidity of the membrane. Cholesterol also contributes to stability of the membrane. For example, at hot temperatures, it prevents the membrane from becoming overly fluid. Another type of lipid in our cell membrane are sphingolipids, which are just 4% of the total mass of membrane. Sphingolipids have a special type of alcohol which is called as sphingosine, as compared to glycerol alcohol which is present in phospholipids. Let's discuss some functions of lipids in the cell membrane. These lipids, especially the phospholipid bilayer, makes the backbone of cell membrane. These phospholipid molecules are just like pickets of a fence around a house. These lipids are also responsible for fluidity of the membrane, which means our membrane is not rigid, its molecules in it are constantly moving. Lipid bilayer is also a source of lipid signal molecules. For example, from some of the fatty acid molecules, we can get prostaglandin, which is an important mediator of inflammation, so which can be used as a communication signal outside of the cell. Some communication signals are used inside of the cell. For example, inositol triphosphate, IP3, are DAG, diacylglycerol. They are important second messengers in the function of some hormones. So we can say that some of the lipid signal molecules can be used for communication with outside of the cell or inside of the cell. This lipid bilayer, this is also a barrier to diffusion of water soluble substances like urea, glucose and ions. Some complex type of sphingolipids also take part in providing protection to the cell from harmful substances and also in adhesion of one cell to the other cell and in signal transmission processes. Proteins are large globular molecules which are interspersed in the lipid bilayer. In terms of number of molecules, proteins are 50 times less than lipid molecules. However, because these proteins are bigger in size, they make 55% of the total mass of cell membrane. Two types of proteins are present in the cell membrane: integral and peripheral proteins. Integral proteins are those proteins which are either partially or completely embedded in the cell membrane. Those integral proteins which completely pass through and through the lipid bilayer, they are called as transmembrane proteins. Transmembrane proteins are more common as compared to partially embedded integral proteins. Peripheral proteins are those small globular molecules which are present either on outer surface of the cell membrane or on inner surface of the cell membrane. Being polar molecules, they cannot pass through the cell membrane. So this is why they are present either towards the intracellular surface or towards the extracellular surface. And they are attached through weak chemical bonds either with integral proteins or with lipid molecules. The protein molecules which are present in lipid bilayer are analogous to freely floating icebergs in a moving sea of lipids. And this brings us to the fluid mosaic model of cell membrane, which is one of the best models presented so far to understand the structure of cell membrane. In this model, fluidity is provided by the phospholipid molecules and mosaic is the ever-changing pattern of protein molecules. Researchers have recently discovered some patches of cell membrane which are more specialized, highly packed and thicker in structure. These specialized patches of cell membrane are called as lipid rafts. They have extra cholesterol, more sphingolipids, and an abundance of a specific type of protein. For example, in the pancreatic acinar cells, such patches of membranes have abundant receptors for cholecystokinin hormone, which increases the production of pancreatic enzymes. These lipid rafts may be flat or may be indented like a cave. Then we call them cavuli. Cavuli are especially important in the cell membrane of smooth muscle fibers. Now let's talk about the function of proteins which are present in cell membrane. Some of the transmembrane proteins behave as channels or pores. These are water-filled pathways. These protein channels are highly selective for the ion which they transport. For example, we have sodium channels which transport only sodium. We have potassium channels which transport potassium. This selectivity of the channels is because of the diameter of their pore, presence of specific groups on the inner surfaces, and also presence of some negative or positive charges on the inner surface of their channel. There are two types of channels: leak channels and gated channels. Leak channels are those channels which are always open for the specific type of ions which they transport. The other types of channels are gated channels, which have gates, which are parts of the protein molecule present either on outer side or on inner side or on both sides of the channel. These gates control the opening and closing of the channel and hence the permeability of this channel. If gate of a channel opens by binding of a chemical, then this kind of channels are called as ligand gated channels. For example, acetylcholine gated channels. And if gate of a channel opens by change in the membrane voltage, then we call them voltage gated channels, like sodium voltage gated channels which open during depolarization of a nerve fiber, and potassium gated channels which open during repolarization of the action potential of a nerve fiber. Some channels can be closed by some drugs. For example, calcium channels can be closed by the calcium channel blockers drugs, and these drugs can be used for the treatment of hypertension and some types of arrhythmias. Mutations in some types of channels can cause certain deadly diseases. For example, mutation in a type of chloride channel leads to a disease which is called as cystic fibrosis. After channels, another type of transport protein is carrier protein, which is also a transmembrane protein. These carrier molecules, unlike channels, do not have a pore. These carrier molecules can transport some larger water-soluble molecules. For example, they can diffuse glucose from outside of the cell to the inside of the cell along its concentration gradient. This is called as diffusion because this is along the concentration gradient. These carriers they can also be used for uphill transport or moving substances against their electrochemical gradient. Then this type of transport will be called as active transport, which can be primary active transport like the transport through sodium potassium pump, and another type of active transport is secondary active transport. We will study these types of transports later on in another session. Cells vary in terms of carrier which they have. For example, in the thyroid follicular cell, on the basolateral surface, we have a carrier which transports iodide, and this is only present in our thyroid gland and not elsewhere in our body. Because different cells vary in terms of channels and carriers which they have, hence they vary in their response to various messenger molecules and drugs. Some proteins present in the cell membrane they behave as enzymes. Some of these enzymes are present on the inner surface of the cell membrane and some on the outer surface of the cell membrane. Different cells are specialized in terms of enzymes which they have in their cell membrane, hence the reactions which they perform. Many proteins present in our cell membrane behave as receptors on their outer side because they respond to the messenger molecules which are coming from the extracellular environment. Some examples of these messenger molecules are water-soluble hormones, for example, insulin, glucagon, epinephrine, norepinephrine. Different cells vary and are specific in terms of receptors which they have. For example, thyroid follicular cells are the only cells in our body which have receptors for thyroid stimulating hormone. So this is why they are the only cells which respond to TSH hormone, and no other cell in our body responds to TSH. Another important function of proteins in the cell membrane is adhesion, and these proteins are called as cell adhesion molecules or CAMs. These proteins are important for adhesion of the membrane with the extracellular matrix or the adhesion of the membrane with the intercellular cytoskeleton. These adhesion molecules or CAMs can make loops or hooks which can protrude from the outer surface of the cell membrane. One type of CAMs is cadherin, which is present on the surface of cell, and they attach one cell with the other cell just like a zipper, and in this way they hold cells together to make tissues. Another type of CAM protein or cell adhesion molecules are integrants, which attach the cell membrane with the extracellular fluid, with the extracellular matrix, and also internally with the cytoskeleton. In this way, they provide mechanical link and they are important for providing shape to the cell. Another important function of the proteins is that they, along with carbohydrates, that is glycoproteins, play an important role in contributing to the ability of the cell in recognizing self and non-self. And in this way, they play an important role in the cell's immunity. Another important function of the cell membrane proteins is that they behave as docking marker receptors, which means that on the inner surface of the cell membrane, some proteins behave as receptors for the docking markers which are present on the surface of membrane bound vesicles, which may be packed with some hormones or neurotransmitters. When those vesicles, which are having some marker protein on their surface, they come and bind with the receptor, then just like a lock and key fashion, this bond is formed, and then the vesicles will burst open and then it will release its neurotransmitters or hormones to the outside of cell by exocytosis. Now let's talk about the carbohydrates which are present on the outer surface of the cell membrane. Carbohydrates are just 3%. These carbohydrates they provide sugar coating to the cell membrane. If carbohydrate chain is attached with the lipid molecule, then we call it glycolipid, and if this carbohydrate chain is attached with the protein, then we call it glycoprotein. Most of these carbohydrates are attached with the proteins, and hence most of them are making glycoprotein. These carbohydrate chains are just like antennas of the cell because they are very important in the cell recognition process, and all these carbohydrate chains present on the outer surface of the cell membrane are collectively called as glycocalyx. Now let's talk about some of the important functions of glycocalyx. Number one, these carbohydrate chains are very important markers for cell Different cells have different types of markers. So these carbohydrate chains, they make a trademark for every cell. For example, our blood groups A, B, AB, and O, they are different from each other because they're having different carbohydrate and protein chains. So these carbohydrate chains, along with proteins, they make one cell different from the other cell because they are different in terms of their antennas. They are different in terms of their cell recognition process, and hence this contributes to the immune reaction of the cell. Because these carbohydrate chains are important in self and non-self recognition of a cell, when the cells will recognize that it is surrounded by some other similar cells, then those cells they group together to make a tissue. And this is very important for growth and development. Moreover, when one tissue is surrounded by some other tissue which is different from it, this tissue, when it is growing and making more and more cells, will not cross the limits of its own natural boundaries. This is normal for almost all the tissues in our body. This is why our liver is confined at a certain place, and spleen at some other place, and the kidney in some other place. But this is not true for the cancer cells, because cancer cells can move beyond their own limits and one tissue can enter into the other tissue or organ. Most of the carbohydrate chains are negatively charged. Hence, these negatively charged carbohydrate chains make the outer surface of the cell membrane negatively charged, and hence this negative charge on the glycocalyx will repel other negatively charged objects like bacteria. These carbohydrate chains, along with protein molecules, also make some receptor sites. For example, receptor for insulin. Now let's talk about some general function of the cell membrane. Number one, cell membrane is responsible for maintaining the composition difference of the intracellular fluid from the extracellular fluid. For example, you know that intracellular fluid is rich in potassium and extracellular fluid is rich in sodium. This is because of selective permeability of the cell membrane. Cell membrane provides a mechanical barrier, and in this way it compartmentalizes the intracellular fluid from the extracellular fluid, and this provides a protection to the cell. Cell membrane also controls the entry of some nutrient substances into the cells like glucose, and it also controls the exit of some waste products like carbon dioxide to outside of the cell. The difference in composition of various ions like sodium, potassium, chloride, calcium is important for maintaining the membrane potential and also for electrical activity of the cell. Attachment of the cell membrane to the extracellular matrix and also one cell membrane to the other cell membrane is important for making tissues and organs. Cell membranes respond to various messenger molecules. For example, it has receptors which respond to various hormones and neurotransmitters. Please keep in mind that the difference in functions of various cells is because of minor differences in the composition of their cell membranes. So it means the difference in lipids and also difference in the types of protein, channels, carriers and receptors in different cells is responsible for difference in their functions. And last but not the least function of our cell membrane is that it is the anchoring site for various components of the cytoskeleton, and hence it maintains shape of the cell. So far, we have studied the structure of cell membrane, which is very important basis to understand the transport of various substances through the cell membrane, which we will study in our upcoming lectures. We have also studied some basics about channels and carrier proteins, which are very important foundation to understand the resting membrane potential and action potential, which we will study in nerve and muscle physiology. I hope you must have enjoyed this session. Thank you so much for watching this video. See you next time with another video.